Okay, in this video, I'm going to go through the stages of mitosis. So that's, hold on, mitosis with a T. So mitosis is part of the life cycle of the cell, and it specifically talks about the part of the life cycle where the cell prepares itself and divides into two. And mitosis has different phases. We move from interphase, which isn't strictly in mitosis, but it is part of the cell cycle. So we go from interphase, and then we go into prophase. Then we go to metaphase. Then we go to anaphase. Finally, we have telophase. And to end it all up, we go through something called cytokinesis, which isn't strictly part of mitosis. It's like the actual division of the cell, but these four make up mitosis officially. And the whole list is really the cell cycle. So in other textbooks and on websites, there's a lot of detail about all these things, and there's lots of like extra in-betweeny phases, like pre-prophase and pro-metaphase, like halfway between prophase and metaphase. I'm not going to worry about that. You don't need to spend time on all those really extreme details. I'm just going to go through these main stages and tell you the kind of hallmarks that you need to know for this class. So it's not as detailed as you might get in some other kind of readings. So I'm going to flip on to the next slide. And I'm going to show you some diagrams of what's happening in each of these phases. I'm not going to write all this down, so this is for you to take notes from, all right? But we'll start off up here, and this is what interphase really looks like. The cell spends most of its time, ooh, that's bad writing, most of its time in interphase. So when you look at a cell on the microscope, it's probably in interphase. You can see the cell membrane. You can see the nucleus usually in the cell, and the nucleus just looks kind of like gray and fuzzy. There's no real definition in there. Inside that nucleus are all the chromosomes, all the DNA for that cell, but they're very relaxed. The DNA is very unwound. It's very stringy and thin, and you can't really see it at all. It just looks like this gray haze. All right, so then during all this time of interphase, the cell eventually copies its DNA. You still can't see it. And then it goes through G2, which is where the chromosomes do get wound up, and they eventually start to become so thick because they're so coiled up that you can start to see them. And that's when we kind of enter what we call prophase. So this diagram here is prophase. And let me check that you can see that. Okay, yeah. So there's three things to look for in prophase. Number one is that our chromosomes start to become visible. And you can see them here. They're these black kind of wormy looking things that look like bow ties. These are the chromosomes. The chromosomes were in this nucleus. They're there, you just can't see them. But when we enter prophase, the DNA is becoming more and more wound up, and we call that condensed, kind of squished together, and it starts to become visible. So that's number one for prophase. Chromosomes become visible. They're replicated. They look like these X's. The nuclear envelope, which is the membrane that surrounds the nucleus where the chromosomes are kept, is starting to disintegrate. And that's why it's shown here in a dotted line, right? See, this is starting to go away. So that's another part of prophase, is the nuclear envelope disappears. And the third thing for prophase is the spindle starts to form. This is the spindle. It looks like some whiskers. What the spindle is a bunch of fibers that stretch from one side of the cell, inside the cell. This is inside the cytoplasm. But the spindle are a bunch of fibers that make this kind of net that spread across the cell from side to side. You can see here they're starting to grow. And eventually they're going to grow all the way across, and they're going to join up in the middle. So you're going to get all these kind of like fibers that are going to grow across. Right now they haven't joined up in the middle yet. They're still being formed. But that's one of the hallmarks of prophase. So once again, prophase, three things. The chromosomes are going to become visible because they're being so wound up. and condensed. The nuclear envelope is going to disappear. So the nucleus is going to kind of, the thing that contains the DNA is just going to go away. And the DNA is going to be loose in the cell. And the spindle fibers are going to start to form. That's prophase. All right. Next, and that's what comes first. Next is metaphase. So I'm going to write that here, metaphase. Hopefully it's not coming off the edge. Oh, I'm right on top of it, aren't I? Let me move myself. Hold on. Then you can see. I'm going to put myself over there. Better? All right. So here we have down here, this diagram here. This is metaphase. Now, metaphase is really easy to spot because I think of M for metaphase and it means M for middle. And in metaphase, all the replicated chromosomes are all lining up across the middle of the cell. Right? It's the only time you'll see them all lined up like this. So you can see that the nuclear envelope is completely gone. There's nothing containing this anymore. There's nothing kind of around this anymore. It's gone. And the spindle has completely formed. So these fibers are now stretching from side to side in the cell. They're anchored onto these little bodies here. These are called centrioles. That's not super important, but the spindle fibers have formed. But what you're really looking for in metaphase is the lining up of the chromosomes, the replicated chromosomes. They look like these black X's across the middle of the cell. And they actually attach, the chromosomes attach their centromeres, which are the little dots in the middle, to the spindle. So each chromosome is like an X connected in the middle. So here's one chromosome, here's the other chromosome, and it's been replicated. We'll get to that next week. And they're connected at the middle, which is the centrosome where my knuckle is. Centromere, excuse me, the centromere where my knuckle is. And the centromere then like attaches itself onto the spindle fiber. And that's kind of what you can see right here where I circled it. So that's metaphase. Everything's lining up. All the chromosomes, replicated chromosomes, are lining up in the middle of the cell. Sometimes this is called the equator of the cell because cells are kind of roundish, and this is across the middle like the equator. All right, step 3 or phase 3 here. This is what we call anaphase. And this one is also very easy to spot. If you could see it, I'm going to write it up here instead. Anaphase. And I like, this one's easy to spot because A for anaphase, A for action. There's a lot of action in anaphase. If you have watched the little video of the green cell dividing that I posted here, you'll see here's my replicated chromosome. I'm going to use my hand this time instead of my knuckles. All right, and it's stuck to the centromere, and the spindle fibers are going this way and this way. All right. What happens in anaphase is the spindle fibers suddenly contract and get very short, and they pull apart the X, the replicated chromosome. So here's my X. I don't know if you can see it very well. It's hard to bend my hands and get it lined up. But my spindle fiber's coming out this side, and I've got a spindle fiber coming out this side. And in anaphase, the spindle fibers tug, 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 and pull the two sides of the X, the two sides of the replicated chromosome apart. So it's very action. It looks really dramatic when it happens if you see that animation. Anaphase is the pulling apart of the two sides of the replicated chromosome. And you get this kind of like V where they're being kind of dragged one from the other. It's leading here. This is the centromere attached to the spindle. Here's the centromere attached to the spindle. As the spindle gets shorter, everything gets dragged back to either side of the cell. All right. Last but not least, we have telophase. Oh, let me spell that right. Telo or telophase. Okay, and telophase is kind of like the opposite of prophase. So all the things that happen in prophase kind of undo themselves in telophase. So what do I mean by that? Well, there's three things going on here. Number one, the chromosomes are starting to relax back out. So they're not quite as short and fat as they were. And that's because the DNA is starting to unwind itself and kind of relax like long threads, whereas rather than being wound up balls. So that's the first thing that's happening in telophase. The spindle is starting to disintegrate. You can see these fibers are starting to kind of break down. And the nuclear envelope is starting to come back. You can see this dotted line has reappeared. But we're actually building this twice over because the cell is about to split into two. And so we have to build one nuclear envelope for this new cell and one nuclear envelope for this new cell. All right, so those three things, the DNA relaxes, the spindle breaks down, and the nuclear envelope reforms to create the two nuclei for the two new cells. That's kind of the opposite of the three things that happen in prophase. And then last but not least, we have this process here called cytokinesis. This always confuses people. Cytokinesis is not officially part of mitosis. Cytokinesis really stands for the breaking apart of the cell. That cyto, cell, kinesis, like coming apart, or separating. And so here's my cell, starts off like this. All this stuff is going on inside. The prophase, metaphase, anaphase, telophase is happening. And then once the cell has two separate nuclei, which is what is forming down here in telophase, once these are formed, then the cell can pinch together and pull apart. And now I've got two cells. And there's a nucleus in this cell with its own package of DNA, and there's a nucleus in this cell with its own package of DNA. And that pinching apart and separating into two, that is what cytokinesis talks about. It doesn't really talk about the DNA and the chromosomes and all that kind of stuff. It's really the pinching apart of the cell into two. And that's the last part of this life cycle. So those are a description of the stages of mitosis and the things you need to look for at each phase. Very simple, nothing more detailed than that. Just look for those hallmarks that we just talked about.